looking at information for your DNA lab. There's a couple little tricky things, and I'll try to highlight them right now. I'm not going to try to teach the whole DNA lesson. First of all, in DNA analysis, you are not sequencing an individual's entire genome. You are not coming up with a 3 billion base pair specific identification for a single individual. What you are doing with DNA analysis is looking at junk DNA, specifically known as introns. In between genes, it's almost like the sequences hiccup. You'll notice right here, this is the same as, this is the same as that. It hiccups and stutters, and it's junk. It doesn't code for anything. But scientists notice that this hiccuping and stuttering is universal to all humans. Between specific genes, there will be this repeating junk DNA. And it is known as a short tandem repeat. And the FBI, when they do DNA testing, they look at 13 specific short tandem repeat areas. And then they use that information to try to match against other individuals. So where did these STRs come from? Well, you know, here's a chromosome. You've seen this before. One of these comes from your mom, and one of them comes from your dad. So, looking at one chromosome, here, for example, might be the one from the mom, and here might be your one from your dad. The gene you inherited for your mom had a hiccup or stutter, that short tandem repeat, that repeated 7 times. However, the gene that you inherited from your dad did it 8 times. Now, knowing this, that you can get some heterozygous variability between short tandem repeats allows to increased specificity and individuality in DNA testing. And I will show that to you right here. Here's another slide showing some differences. Now, you'll notice here, some of you may go, hey, we're looking at 4 base pairs now instead of 3, and that is true. The stuttering has different lengths for different short tandem repeats. So maybe the one on this chromosome might stutter in 4 codons. Maybe the one on this chromosome will stutter in 3 codons. But that is consistent through all humans. And I will try to explain that better when we look at which specific genes we're looking at. So the number of repeats is highly variable. For some genes, they may range anywhere from 4 to 40 stutters. So one individual may inherit a short tandem repeat on their one chromosome that stutters 4 times from their mom, and it might stutter 40 times from their dad. Okay, so looking at specific, and this is, you know, rough evidence that comes right out of the machine. Okay, this is what it looks like. Now, I'll have a close-up here in just a sec. These are the different genes, and you can't quite see it from here, but they have very specific names. This one I think is like DS36138. I don't know what gene DS36198 codes for. Maybe it codes for eye color. Maybe it codes for the enzyme that helps you digest carbohydrates. I'm not sure. But what I do know is when you put that through your DNA machine, you're going to get a variability right here on this axis of the graph. You'll notice here it looks like it ranges from 0 to 20,000. The higher the peak, the more times that gene stutters. Okay, so obviously this individual right here, we're looking at one gene, Okay. And since there's 2 peaks, this person is heterozygous. Yes, that means they one of their genes from one parent did not have quite as many stutters as their other parent, so it's not going to come through at the same time. And here I have like a close-up. Just a sec. Okay. So this axis right here is just an indication of, and it has to do with fluorescence. So we're used to reading this axis and going, oh, that's the information we want. Because when we look at, time temperature graphs from science or whatever, we go, oh, we're coming across this way to see at what time, and then we see the quantity of the temperature. Oh, it's 90 degrees at this time, it's 80 degrees at this time. For our intents and purposes, we're not so much worried about that. We are actually worried about this direction on the axis. I know, and that's a little bit different, and that's where students get hung up on this assignment when they are completing it. So we have 2 peaks. That means this individual from this DNA test is heterozygous for the D19S433 gene. Here, this has the x-axis on here has to do with the weight, or the size of the particle, I should say. It's more dependent upon size than weight. And because this one right here came through earlier, it is smaller, and it's been calibrated that this one right here, if it comes through at this point right there, it has 13. It's repeated 13 times. It stutters 13 times in that section of junk DNA. Now this one that came right after it, because it is along further on this axis, it is larger, and it actually stutters 14 times. So this individual is heterozygous for the D19S433 intron 13, 14. However, this individual looking down here at the TPOX gene, notice only 1 peak. That means this individual is homozygous. It inherited 11 intron stutter sections in its genome from mom, and 11 stutter section intron genome base pairs from its dad. Okay, so that's a little introduction. Now specifically, that's stuff we would have gone over if we would have had the class. This is the 13 different DNA STR sections that the FBI gathers data on and uses to identify individual. There are more than 13. You'll notice there aren't even any on this number 1 chromosome. Okay, there are more than 13. This is just the ones that the FBI has selected, and that's just there for your gee whiz if you want it. Now, specifically for your lab, okay, when you are working in the lab, everything is going to be available within the lesson. You don't have to start a virtual lab or anything like that. What you're going to look at is some DNA evidence coming straight off the machine, like the one that I showed you over there. This panel right here that I selected happens to be the page where you're looking at DNA from victims, and right now I have it highlighted on victim 1, so you'll need to click on victim 2 and victim 3 to look at their information also. Now again, this right here has to do with the reflectivity, reflective units. You don't need to worry so much about that. You're not going to go, oh, well, this one is higher than that one, so that means, no, you're not going to do that. What you are looking at is how far along on this axis it is. Okay? Looking at the red D3S1358 gene, that's this right here. That first peak happens about right there on the chart. That second peak happens about right there, and I wrote them so close together that the computer is not going to be happy. So both of those are between 110 and 120. Okay. Now here's the part that a lot of students don't notice. You need to click this button right here and this chart pops up. Okay. What you're going to do, since we're looking at the D3S1358, you're going to look at D3S158. You're going to look at, did we have 115, a peak at 115? Yes, it was. And we also had a peak at 119. That correlates with a short tandem repeat of 11 and 12. So victim 1 has a D3S1358 of 11, 12. Okay, now, and I put this so it covers it. The other one that I did is the D18S51, and that happens to be this blue peak right here that got covered. You'll notice that it peaks right there, 260, 270, 272. Reading this along the bottom, this line right here would have been 270. It's just over that, so 272, because I know it's not 268, I know it's not 276. What's left? 272. Coming across there, that correlates with a short tandem repeat allele number of 9. It hiccups 9 times. This D18S51 short tandem repeat hiccups 9 times. So you'll want to look at the DNA data for each victim, look at each of the different alleles that they present you, and then keep track. So you'll have a chart, something like this right here. Now this chart right here only has one evidence sample and 2 suspects. And in each of our cases, we're going to look at 3 suspects. But create some kind of a chart like this with the STR locus, the gene that you're looking for. Then you can compare the evidence sample to the different suspects. And in this case, our evidence matches which suspect? Suspect B. See, 1517, 1516. 2327, it matches all the way down. If you have a match all the way across, then you've got a match. Suspect A, it doesn't match. I don't think it matches with any single one of them. So yeah, we know it is not suspect A. Larger tables might look something like the one up above, where you have several different categories, descriptions, and you've got their genome off here to the side. So that's what you need to do with this lab, is to analyze all of that DNA data, be able to compare it, and then make conclusions based on what you find comparing that DNA data. And as always, here's something to smile to get you on your way.